Okay, thank you. Welcome everyone, and it is a great pleasure and true joy to welcome Professor Matthew Collins, who will deliver this year's CENIEH Distinguished Lecture. Matthew Collins is Professor of Biomolecular Archaeology at the Globe Institute, Faculty of Health and Medical Science, Deputy Head of the School of Archaeology at the University of Copenhagen, as well as Professor of Paleoproteomics at the McDonald Institute for Archaeology Research at the University of Cambridge, if I got it right. So I could say that Professor Collins is among the leading global experts in the fast evolving field of paleoproteomics, but I will go further and say he is quite simply the best. So Professor Collins combines extraordinary scientific expertise with a brilliant and creative mind. But I will say that what truly sets him apart is his generosity, his willingness to share his knowledge, his time, his ideas, and it's this spirit of openness and collaboration that is really drive the field forward. So I would also highlight his unwavering commitment to critical thinking. In times where scientific enthusiasm often races ahead of methodological caution, Professor Collins reminds us that true progress comes not only from discovery, but from doubt, rigor, and courage to question. So his presence here today is particularly meaningful for us as we embark on the exciting journey of launching a new line of research and dedicated laboratory in molecular paleobiology at CENIEH. In a field that is still taking shape, voices like his are essential, not only because of their vision and expertise, but because they embody the hope that paleoproteomics can grow anchored in scientific integrity and intellectual depth. So we are also immensely grateful that Professor Collins will be teaching a week-long workshop during his stay, an extraordinary opportunity to learn, grow, and train the next generation of researchers in this emerging field within a philosophy of openness, curiosity, and critical reflection. So we look forward to listening to you, learning from you, and sharing this moment of scientific exchange, and hopefully to collaborating with you in the near future. A single conversation with you is enough to recharge any spirit minimally sensitive to science, biology, and the wonders of evolution. So thank you very much. It's a true luxury to have you here. Welcome.
It's very kind. Don't clap for me, clap for that speech. The rest of it may be downhill. So yeah, I was reflecting on what to talk about today, and I was really thinking about the question of dinosaur protein. And the reason for that is because this is something where we're reflecting at the moment on. There's a new version of Jurassic Park out, Jurassic World. You know, we're moving forward. We think about, well, what would life really be like if that was possible? And you kind of come to this, right? We're in this very, very strange world now where some of the ideas that people discuss are kind of, beginning to happen in strange ways, which I would argue often are slightly overhyped. And so what I want to do is really think about the way that proteins has changed over time, and then how you, particularly here, can benefit from sort of the new research that's being done. And the first really simple point to make out is that this, this is the amount of DNA required to code those 6 peptides. So look how much more compact the information is. And because of that compactness, because we've got 20 different amino acids in this message, it's a message that is more compact, it survives for longer. And that's clearly important here with the very long records that you're studying, and also because this is a molecule which survives as a consequence of time and temperature. The colder it is, the longer it survives. And whatever you lot say, it seems to be pleasantly warm in this part of the world. So that's a challenge. There's 2 ways that you can go about studying really old proteins. You can dive in to the deep, or you can slowly wade in from the shallows. I'm a wader. I'm very cautious. I don't believe anything. And so that's interesting when it now comes to reflection on 33 years since Jurassic Park, 25 years now since the first, 20 years since the first dinosaur collagen sequence. We'll talk more about collagen in a moment, that was reported. And I was on Blue Sky actually saying now, you know, I've always been hypercritical of these kind of claims, but maybe as this was an independent lab that repeated the work on dinosaur collagen, maybe if we look at the data carefully, it may be true. I'm not convinced yet, but things are moving. And so I think that's really intriguing. And there's a question down here that you can't read, which says, how long will collagen last? Now, the person that knows the answer to that is sitting in this audience. It's Colin Smith, because he did some really careful research a long time ago where he calculated experimentally, in a set of experiments which took him almost 3 years, I think, to generate the data, how rapidly you lost collagen from bone. And then he went and searched across evidence from radiocarbon dates and was able to show that the model he had made in the lab, this blue line, did not fit at all well bone, apatite, and charcoal, other materials that you can date, but almost it was uncanny how well it fitted the survival of collagen. So this is taking two different types of data and demonstrating it is predictable to study the survival of collagen. And as you notice, this is high temperatures here, not good preservation, and so we get poor survival. And that's great. And I thought this was the best quality evidence we had of any kind of degradation. And then, of course, what does Carly Peters come along and do is she finds a site in Australia which completely breaks that limit, which shows you that, yes, it's broadly true, but it's not always the case. So we really have to always think critically. Don't believe, keep searching for the evidence. And we do not know why, at this site, collagen is surviving so much further back in time, but I really hope someone studies that site, because this is meaning in tropical and subtropical sites, we're getting long-term persistence of collagen, and I'm fairly sure DNA, and if we can identify those types of sites, we can go back even further. So, and then, this is an email that came in yesterday, saying, yeah, would you talk about 2 papers coming out in Nature with ancient proteins to 18 to 20 million years? So, this idea of this extended timescale for proteins is becoming more and more acceptable, even if I said it wouldn't survive more than 1 million years. So, I was wrong, which is nice. I want to be wrong. Here, I don't want to be right. And so the next question is, but how do we know it's really there? How do we know we've got ancient protein? And so that's a really challenging question. And it's all about authentication. And authentication is something that I know Sam is also really interested in. Here he's leading the New Proteins Lab. But it's a really difficult question. And so this is a set of criteria that we can use to say, we've got a bit of ancient protein. Is it really real? And we can say, could another lab independently replicate it? And I think that's very helpful, because labs have contamination. I can tell you stories. We can say, does it behave in the way we expect those molecules to behave? Like, as you saw, I would predict collagen wouldn't survive in these sites in Australia, and it did. So yes and no, molecular behavior is useful, and there's various kinds of information we can look at, and whether the associated remains make sense. Now that's a good list. I like that list. That is a very old list. That is a list from the year 2000, in the early days of DNA. And it served the DNA community well. And so it's the kind of thing that we should be thinking about. And I think we should be revisiting the successes and the failures of ancient DNA when we move forward with proteins. Let's make sure we do the right things and not the wrong things. And so an example of that is there's been 2 prizes for Nobel Prizes. First one was on radiocarbon dating, and the second one has been on ancient DNA. Now, why was that prize awarded for ancient DNA? There was one primary reason, it became possible to distinguish between modern contamination and ancient samples using a pattern of degradation, which is these young researchers, these new young communities which are emerging. This is the SPAM community. They've made the little book of smiley plots. And here are the smiley plots of DNA damage. If you have a big smile, the DNA is damaged at either end. If it's flat, it's not damaged. And so because of that, Svante Pääbo was able to identify Neanderthal DNA because of this damage pattern developed by Adrian Briggs. And as a consequence, the field of ancient DNA has grown very rapid. You cannot see this graph, I'm sorry, but this is from something early to 2024. This is a logarithmic scale, which you definitely can't see, and that is 10,000. There's been 30,000 articles in Google Scholar with the words ancient to DNA one after the other. And then here we have paleoproteomics, which is clearly much, much younger. But the point is, the acceleration is similar to here. We've got a field which is beginning to grow, and we need to make sure, as we get to this inflection point, that we make the right decisions in our discipline. And then the question is, how do we know we've really got ancient proteins? And I want to focus on one of those things, on a thing called the molecular behavior. The behavior of those molecules. And we have been spending a long time recently, have papers on the archive, studying a thing called deamidation, which is like the deamination of DNA, but unlike that lovely smiley plot, it's not at all smiley. It's really hard to make any sense of it. And this is quite a sophisticated model we put together, where it's on the archive, and what we're trying to do is predict deamidation based upon the physicochemical properties of the individual building block, the amino acid, within the environment of its protein. And when we do that, we get really miserable data. What we're finding is that all the characteristics we wanted to use, which is to do with chemistry, and there's a long list of them there, you can read in the paper, don't really work very well. The main reason you could detect the mechanism of deamidation in our study was you had a good quality spectrum. In other words, it was not about the chemistry, which it really is about, is could I see it or not? So that's a really bad sign. It means that we need to get better in our mass spectrometry at characterizing these things. So deamidation is kind of difficult. And, he says, clicking forward, so one thing we've been discussing about today, when I was at the site, was a thing called amino acid racemization. So all of you can think about the fact that you have a left hand, levo, living. Amino acids are handed because they have 4 different components coming off that central C, which is called an alpha carbon. And they can either be in the left or levo form or the D-dextro dead form. And what happens over time is all the amino acids in your proteins are made in the L form, but they slowly decay and become a mixture of L and D because the D form then switches back to L. This is a really old-fashioned technique. It's been around for years. But I think it's really appropriate as we move forward with ancient proteins. And why do I think that? Well, here is an example of us measuring the racemization of different amino acids. They have different rates. Some are fast, like aspartic acid here. Some are very slow. This is 3 million years in an eggshell. And it's actually not in the shell itself. It's in this little tiny cap. This is a tiny thing, so that's 1 millimeter scale bar, which is made of calcite. The shell is made of aragonite. Aragonite is a kind of form of calcium carbonate which is not stable. So over time, that system doesn't work, it leaks. But the calcite is robust, like eggshell, and like tooth enamel, it doesn't leak, and it gives a reliable clot-like behavior. And what that enables us to do is take lots and lots and lots of sites. These are all the quaternary sites in Britain. And where you've got a coloured symbol, there's over 120 sites here, you have lithic archaeology. And what you can see here is that we've plotted 2 parameters, what's called the free aspartic acid here, and the total hydrolyzable aspartic acids here, this is a thing called alanine, and they should always fall in a line. That tells you it was behaving as a closed system. So you've got internal quality control. And then you can see, you can just read off. We've got Mesolithic stentils, we've got Upper Paleolithic, we've got Levallois, we've got a Schuling, Clactonian round down to Mode 1 in Haysborough. But what's interesting, there's nothing here. That's great. There's no lithics in stage 5. And so we're able to tell just from rastomization, we can do the chronology and we can explore, and in fact within that we've got within substage resolution. Right? That's in calcite eggshells of gastropods, which you almost certainly have not got at the site. But what you have got at the site is tooth knot. And so think on tooth knot. And what can you do with it? Well, Giff Miller, who's a geologist working in Australia, can demonstrate some of the lovely things you can do with it. So here, for instance, he's measuring rastomization, and this is fully rastomized eggshell. This is going back, it's Australia, it's much hotter, 140,000 years. But here's the line of data he's got, and notice it's not a straight line, it fluctuates. This is the end of radiocarbon. Now we're into using OSL and U-series. The point is, it's not a constant rate, it fluctuates because it's a molecular reaction, not an atomic reaction like all the dating techniques you're aware of. So it's more complicated to do. But once you have got good chronological control or good paleoclimate control, one will work with the other. So I think it's something that we should, or you guys should think about here, rastomization, tooth knot. And what gets me is, despite the fact when you look at it from a chemical standpoint, it's an amazingly efficient and tool, it's hardly ever been developed. So this is over time, it has never ever been used widely. And it's never been used widely because no one has bothered to isolate a closed, stable system where the chemistry is predictable. But we can do that in tooth enamel. So we have a lot of teeth at the sites here. This method can be applied. Okay? And it's what's really neat is you get for every single sample a date, and it's a relative date admittedly, but you can also do all the other chemistry on that same sample. So you get a date and the chemistry, and this is Giff showing how he's plotting carbon isotope values against the date based upon the rastomization. So you're working on the same tissue, and you're getting more information from it. And so we then have to think, as we get very excited about all these new papers coming out, but is that real? How good is proteomics really at telling stories? And so we have, this is my ovicaprid, a thing that doesn't exist. It's not really a hybrid at all. It's a functional term used by zooarchaeologists because they can't often morphologically tell sheep from goat. And it's the reason that we developed zooms originally, to tell apart sheep and goat. So let's look at this data set. This is a really important data set. There's been a whole series of these data coming out now, using proteomics to go beyond the limit of hominid evolution. And this is a sample from Harbin. Okay? And if we then look at that data, look at it closely, something might happen on the screen, yeah. I've got two data sets you now have to think about, okay? We've got a proteomics data set from Harbin, and we've got a genomics data set from Denisova, okay? And what we're looking at here, these are letter codes. K and R are different amino acids. There's a K and R here, there's a model R here, there's a G Swappington R here, there's an E to G, M to G. So these are different amino acids that are changing in Harbin. These are different amino acids, it's R to a G to an R, and H to an R. Different amino acids changing in Denisova. But you might notice that in all these other things, there's no changes at all. Okay? And that's really great. What that means is that proteomics has picked up variation in these samples, which is allowing to say something about human evolution. But, as in the introduction we heard, you have to stand back. You have to think about the data. Don't be uncritical about the data as it's generated. Let's think about this, not in terms of have we or have we not got Denisovan, have we or have we not got a functioning collagen molecule itself. If the molecule of collagen can't exist in biology, those sequences can't be right. Okay? So let's think about that. And now you all have to learn a bit of collagen chemistry. So I hope you don't mind. So this is the molecule collagen. It's weird. It's not a normal protein. It is utterly abnormal, which is why it survives so long. Incidentally, you guys should be thinking about getting near-infrared because it's got weird near-infrared characteristics which are very useful for rapid screening. And you can sample to a depth about 2 millimeters in a bone. I was actually going to put, we just had a paper, we've just submitted it. Anyway, but the point is that work from Tina in University of Colorado. So 3 chains, 2 of one, alpha 1, and 1 of an alpha 2. They self-assemble from this end. They roll themselves up into a chain. Then this bit gets cut away. And then they self-organize and have a series of crosslinks. And then they form a fibril. It's a molecular rope. It's a weird, highly orchestrated, highly complex structure. I'm going to show you some movies what I made. This one is how it melts. This one is not one I made. This is how it will self-assemble. And then this one, actually, I'm not going to show you now, which is the beautifully intimate association between the tiny crystals of here, mineral, and the collagen is sitting in the middle. And this is important. Collagen in bone is not hidden from the outside world, unlike enamel. In the case of collagen, it's mineralized, but it's in a mineral cage. So remember that it is part of an open system, a leaky open system you cannot trust for diagenesis. It's complicated. Okay, so let's look at we're just briefly going to introduce collagen. So this is actually skin, but it's kind of bone-like. We're going to go down in scale. Like skin and bone, they're orienting these fibers, these big lumpy fibers in different directions. And here we have the scale bar going down here to smaller and smaller scales. Let's look at one of these molecules. It's really complicated. It's made of multiple fibrils bound together, and you can get down to a final unit cell of 5 of these things. But each of these, and they are interacting, they're lying together because of charge, positively charged negative charge groups lying together. You can take one of these things out. It's made of 2 alpha 1 and 2 alpha 2 and 1 alpha 2 chain. Okay. If we then look at how it melts, this is our molecular dynamic simulations. This is collagen melting, but it's incredibly, incredibly rigid, which is the main part of the story here. And we can eventually begin to get hydrogen bonds, which are very, very strong bonds holding this thing together, beginning to come apart. It will basically pull itself apart by a process of melting. Because of, I want to show you one more movie first. Yeah, this movie first, we'll go back. This is taking those single chains of collagen, those triple helices, putting them on mica, and they start to, that big, big chain you saw, all those chains together, it automatically self-assembles into this ginormously big chain because all of the charge sites, you have negatively charged groups interacting with positively charged groups, and it just forms this beautiful structure on its own. It self-organizes. Okay? So, what that means is we have this wonderfully architected structure. But what it means is to make that happen, the charged groups, which are K and R, remember those letters, R for arginine, positively charged, and K for lysine, positively charged, and the negatively charged group, aspartic acid and glutamic acid, have to be in register. They're little magnets. They can interact with each other. Okay, and so here, this is actually, I pulled this picture off because I'm writing some, I was just showing Sam, I'm writing a program to basically simulate all this kind of working together so we can actually look at degradation in this higher ordered structure. So this is some Python script on Google Colab, which you'll be able to get access to hopefully pretty soon. Anyway, yeah, and it didn't work. We tried to run it this afternoon. It doesn't matter. But, so we now look again at this data, and we say, wow, it's really weird because if you look at Zoom's data, all the Ks and all the Rs in mammals are always in the same place. They never change because they can't change because it has to self-organize. But what have we got here? We've got Ks and Rs. K to R, that's okay. G to R can't happen. R to G can't happen. G to R can't happen. H to R can't happen. These are things that can't happen biologically. Okay? So how are they there? But it gets worse. The other thing is, notice how few alternative sites we've got here. Here we have massive degrees of alternate sites. You get one copy of a gene from your mother and one copy from your father, and it is entirely possible that they could have a mutation. Mutation in collagen, not very likely. One mutation in collagen. This is a list of all of the pathogenic mutations in collagen. We know where they are. Collagen does not like having mutations because it disrupts the folding. And what I was showing in that video, in the program I'm writing, is we're calculating all the melting temperatures and how these substitutions impact on the way the collagen gets built. Okay? So you've got not only massive amounts of heterozygosity in a gene which there's no heterozygosity in, maybe one site very occasionally, 3 in the Denisovan in the sites they found, 50% in Harbin, 50% actually is looking most of the sequences you're generating. There's far too much variation in the sequence to be biologically plausible. Okay? We just know it can't happen because you couldn't self-assemble those proteins. And it's obvious biologically, and you can look at it. These reasons they're there is because these people are getting osteogenesis imperfecta, the fetuses are dying at birth. This is a serious disease. And by the way, some of those mutations actually impact on skeletal morphology. Right? Because they're making your bones, they're making your joints. And so why? Why the problem? Why the problem? So what we've been doing is using a technique called de novo sequencing to sequence collagen. And here's a bunch of collagen. This is, I can't remember which sample this is. Now, this collagen has to have a glycine every third position to make that very tight structure. So I'm just going to take the collagens out the way so you actually can sort of read this better. So this then is the glycine should be there and there and there. And there must be always a glycine every third position, okay? Notice here that the ends of the molecule, it's all pretty consistent. Mass spectrometry does a fairly good job of getting sequences right at the end of the sequence. Oh, we've got a K to an R over there. That's a bit odd. That shouldn't be there. But what you notice in the middle, it's all kinds of nonsense, and the glycines are missing. You saw that in some of the data. It's because the mass spec software is not good when it doesn't know what you've given it. And in this case, because it's what's called de novo sequencing, it's trying its hardest to make sense of little data, and it's giving an honest estimate. Unfortunately, it's not the fault of any of the searchers. They were very fine papers. All these are good papers. All the searchers are very good. They're let down by the software. But you mustn't believe the software. You've got to be hypercritical. And if you want to be critical, it's why didn't they stand back and just think about the biology? Okay? So, okay, what actually can we do with ancient proteins? So, of course, we can do zooms, and this is blowed up. So here we've got all the R's and the K's. So note there's 3 sites here with R not showing, and also I think there's something else not showing, because these are fish, okay? But basically the R's and the K's have to be in register in all these mammals. And they're not in the Denisovan, and they're not in the Harbin individual, and they have to be. So anyway, the point about zooms, which, and don't talk to me, talk to Sam about zooms, but it's a way of doing fast fingerprinting, as well as other things. And the crazy thing is, if in the Harbin individual they'd done zooms and not done sequencing, they might have seen a mass shift they could have interpreted. But they tried too hard to get information out, and they saw too much variation that wasn't there. So, well, what gets me about zooms is it's not the technology, it's how you apply it. And this was a meeting that was held last November in Paris, and the next meeting is here in a year's time. And what really impressed me was the brilliant way that young scholars had thought to use this technology to answer challenging questions. So I think this is something where the community is working really well together, and they're now starting to put forward things like in lab standards and checking out how other labs work. So it's a really great technology, but it's not the technology, it's the minds behind the questions. And so here's an example. This is eggshell. No one can work much with eggshell. But now with zooms, you can easily identify eggshell. And so now Beatrice and Marky's got an ERC project exploring exploitation of birds, because of course they're affected by climate and things. And of course, I love eggshell. It's another closed system. We can date it. It doesn't go far back as enamel. The rates are faster. And then the thing to me that really struck me, we're chatting about today, is just what a gold mine teeth are. And you have a few teeth here. Yeah? It's, there's so much that you can get, because within the tooth, immunogenin, the protein that makes the teeth, is coded on both the X and Y chromosomes. So you get sex information. I think if you want to argue that the Denisovans in China are real, I would do that on the basis of the enamel. I think the enamel proteome looks pretty good. It's the collagen which is so hard to do because of these funny characteristics. So I'm not saying Harbin is wrong at all. I'm just saying that the collagen data has to be wrong. Okay? The second thing then is, of course, with teeth, it's a great system because you've got your isotope scientists, you've got your zoo archaeologists, you've got people looking at wear patterns. I mean, everyone can work together on teeth, and you have a lot of them here. And for me, they're a closed system which allows you to get dating evidence as well. And of course, you can then look at, for instance, with animal teeth, are they hunting male or female animals? And there's so much I think you can do with teeth. I say, from my perspective, the Neolithic and later, a flock is in a tooth. You can read so much about animal management in a tooth. You have to be careful, of course. You can't blindly accept you can do this, so you have to read the literature. And what you learn is that it's not always the case that males carry the Y marker. And this has been well known in forensics. There's many people who are identified as female when they actually were caught, they were male, but they're male that didn't express the Y or had the Y drop out. And this is particularly important in small populations. And so in horses, for instance, it's a real known population problem because of the way that small horse groups have been managed and they've lost the Y marker. So, the literature is all there because it's been used in forensic. So we have to be careful. Don't just say we can sex everything. Check on how challenging it might be. Okay? But also, within teeth, if you're lucky, and it depends on the kind of teeth you've got, you've also got dental calculus. Dental calculus, you've got phytoliths. You might even have starch grain surviving. And this paper, it's not so relevant to you, but this kind of blew my mind. This is from Tina Greenfield and colleagues. They were looking in Mesopotamia, and they're getting proteins from a whole series of plants trapped inside the teeth of donkey and sheep. And if this is true, we have a really rich source of looking at plant exploitation, which we didn't think was possible. Now, I don't think for you, it's going to go far enough back in time to be relevant. But it's another thing that you can do with teeth. And another really interesting thing is teeth of humans, not some animals like carnivores, they form quite quickly, but animals like horses are growing continually. We can then look at the protein pattern of diagenesis alongside isotopic evidence where we get it. And this is some beautiful work on the Irish famine by Julie Beaumont and others. And here, this poor boy dies at the age of 13, and you see changes in his carbon and nitrogen isotopes because he's starting to consume his own body tissues. And at the end here, you get an uptick, and this is really sad to read. So this is a report of, I recommend the person is a deserving person to receive a certain amount of Indian meal. Indian meal was maize, and that maize meant you had a very strong C4 signal here. This was what they fed to horses and animals. But if you were deserving in the Irish famine, you could be given some, even if you died. But that's denting, and that's going to be hard. This paper's come out, and it's really interesting. This is a team that was working together, a team from California working with a local group from this region. So this was good science that apparently everyone's working together. And what they found in enamel is something which is curious. They started finding in the enamel. Remember, the enamel is a closed system. This you can go for. It'll be there. They started to find elevated levels of serum proteins and immunoglobulins, which they argue are related to stress and health. So this will be really intriguing, particularly if you've got continually growing teeth, And you've got that in some of the animals. You have the herbivores with hip slot dentition. So could you get access to that proteome in the enamel and potentially also metabolome, link it to things like enamel hyperplasia and other indications of the teeth failure, and start to explore signals inside the tooth enamel? We've come back to teeth again. And so the last thing I want to talk about is the use of AI, because we've been using AI a lot in my research. And so what we've been doing, I showed you that collagen data, we have all the kind of messy results in the middle, because what we don't want to be doing is using sequence data. We want to be looking at what is there. So this is a project I was doing in Bordeaux with Benjamin Dartigas and the bioinformatics team there, looking at using different de novo sequencing platforms to sequence the proteins. And what we've done then linked it to a thing called OpenSearch, and there's all the stuff we're using is OpenSearch work. And then one of my students, Ishmael, has pulled this together, which is our new strategy. We do de novo first, then we do an OpenSearch, a narrow notch search. We put it all together, and that's how we feel more confident that we've got good protein data. But we can't buy that off the shelf because no one is interested in the kind of problems we've got. So we've had to make it ourselves. So we've got this already. It's on the archive. You can run it. It runs on Google Colab using this strategy. Okay? So we're trying to make all this open so you can use these new software tools to explore this data in a non-biased way, and hopefully we'll get to see better data out of, for instance, collagen. And then another point, and I was talking to Sam about this, is at the moment, though, here's a bit of bad news. The de novo sequencing software using, which uses things called large language models, and you're probably aware of things like ChatGPT, they do a very bad job with what we call PTMs, post-translational modifications. So they're good at identifying the right sequence. This is methionine oxidation the easiest. They can do that quite well. Here's the beloved deamidation. They're doing it very badly. So at the moment, these tools are not good for modified and damaged software, which is why I'm writing new software to find those kind of molecules and then train a new generation of de novo models with that software. So yeah, in future, I think you're part of IRIS here. We're not, yeah, okay. So thinking with IRIS hats on, you know, it's innovation thrives with collaboration, and IRIS is one of those initiatives. Paleoproteomics must be rooted in accessible, low-cost technologies and data sharing. And then we should be really conscious of the ethical and cultural considerations of any work that we do. And this is where I go back to that paper on authentication from the year 2000 and ask us all as a community to reflect on what happened in ancient DNA and really say to ourselves that, you know, we want to make sure all our data is open. That's one thing that DNA does very well. But on the other hand, we are now looking like we can go further back. So this is, I think, what's going to be announced in Nature next week, but this is already on the archive. So this is from rhinoceros teeth, and this is 24 million years with an intact proteome. But yeah, so integration is the key to me. They're really exciting, but you want to work together with your geologists, your archaeologists, and we need to work together. That we need to integrate analytical approaches, of course, because of that. I think there's great potential for me in archaeology and to lead our own methodological development. So I think what we can do, species ID, sex definitely, environmental reconstruction via preservation, yeah, I think so, because of rates of degradation. Diet reconstruction, yes, and if we can get down to whether you're going to get enough food proteins in any dental calculus. And then perhaps, and this is really interesting, there's a paper that's being reviewed at the moment which is saying they actually can do proteomics on lithic artifacts. And I've never been convinced by this, but the new technology looks smarter, so maybe. But I think the main thing is that we have to be really thoughtful, and this is my final slide, that we have to be thoughtful about what we're doing, because what's happened, my hat has been a hat of the acting director of the Duckworth Collection a year ago. And I was really conscious on how these big research groups were coming in and demanding access to material. And we in Cambridge, we had enough staff we could say no, we could ask difficult questions. But what has happened is this is a community where there's been such focus on individuals and small research groups. It's led to a lot of museums saying, no, you can't have access to material. It's not a problem for you, but it's a problem for a lot of other young researchers. And this competitiveness has led to this real kind of toxic atmosphere in the world of DNA with these big lab heads all chasing each other. And I think we really have to be thoughtful about this and not let the same mistakes happen in the world of ancient proteins. Yeah, and with that, thank you all so much. It's been an enormous honour to speak to you all, and thanks to all my wonderful team in Cambridge and Copenhagen. Thank you.
Thank you so much for such a wonderful and exciting talk. So there is time for some questions.
Any comment, question? Hi. Maybe it's kind of naive question, but like, is it possible sometime, maybe not near in the future, proteomics can be used like to retrace to DNA, like with the retrotranscription, to study differences in DNA between species, or to search for DNA that distinguishes one species from the other?
Yeah, I mean, I think it's a really good point, and I think that, of course, the problem with proteins is the DNA has 3 base pairs of codes for 1 amino acid. So we lose that third position all the time. And then the other thing is we lose out on all the other wonderful information that is present in the DNA, because we don't see the intron information, we don't see the structural DNA. So yes, we can see something, but I don't think we're going to see very much. I think that the protein is, you saw how simple and short that message was in the molecular structure. That is a great strength for survival, but it's a weakness because there's not so much information. Now, clearly, we can get sequence data out of these old things, and we've used it to sequence, for instance, a group of extinct birds that evolved 70 million years ago and went extinct 50,000 years ago. And we could say approximately where they fitted in the phylogenetic tree. But honestly, the proteins weren't that useful. And we had an exercise with a wonderful bioinformatician, and she said, okay, let's pretend we have every single protein expressed in that eggshell. And what we saw is that even with all the proteins and no DNA, we still couldn't resolve the tree of birds. So in other words, yes, we can get some way, and as long as we choose the right questions, and I think that's the key thing, there have to be questions which are very, very local questions, sort of population level questions where you might get a mutation in one individual and then that might be shared in the group. So because the problem with the proteins is, I mentioned that you had this R and you couldn't change it. You could swap it out for K because they had the same charge, and there was an example of that, and that's quite common. And that's always happening. The thing is the R will go to K and the K will go back to an R again because that functionally works that protein. And there's therefore a danger if you go too far back, you say, oh, they're both R's, there's been no evolution, but actually went RK back to R again. So the proteins have got some information, but I would say not enough for the kind of things you're hoping to do, sadly. Yeah.
Thank you. Thank you so much for this fantastic talk. We have a journal club in the paleobiology division, and we recently read the Harbin paper and completely missed your point about the biology.
Yeah.
So I think it's a wonderful, so thank you for showing that, because it reminds us that we have to collaborate across expertises in order not to miss those. So I have a question on a different topic about towards the end, when you're talking about the need to not make the same mistakes as ancient DNA.
Yeah.
So, I'm old enough to have been around where we keep talking about needing culture change, but how you actually do that is often really difficult. Yeah. And I'm wondering if you have thoughts or ideas or very specific action items that can be taken to accomplish that goal or to try to?
So what I've done so far is, first of all, I've stood back from all of my roles. I was in a thing called ISBA, and I've kind of stood back, let the young people take over, stood back from my journals except for PCI because that's open access, Diamond Open Access, and then that's for everybody. I've been to visit the head of a thing called Proteome Exchange, which is where they share all the data, and I've discussed with him the needs of our community. And he very thoughtfully said that what we should have done is written a white paper. So I wrote an outline draft and then gave it to this young community called PASTA, which is now one of these whole series of groups that have all been set up. SPAM is the one on metagenomics, set up by a guy called James Fellows Yates, who's wonderful. He comes out of the Max Planck in Leipzig. And then he's helped everyone. PASTA is the proteomics one. There's one called HAM for human ancient DNA. There's one called LARD for lipids. Now, these are all run by young people. And that group have written the white paper on paleoproteomics. Not me, they've written it. And it's come out in PCI archaeology because it's about diamond open access. And I think the thing is, it's giving the power to the new young community is what I would say. And so it is supporting initiatives like the PASTA initiative. It's getting them to write white papers. We're now discussing, the Zooms community are now themselves initiating sample sharing for quality control. We've been discussing, again, this young community about metadata standards. So basically for me, I just do now everything by sort of contacting people in PASTA and saying, I think you should do this, and I'm there to help. My name is on the white paper, and I said it shouldn't be, but they wanted some senior people on there, so 3 of us are on there as well. But I think it's all about giving the future to the young community and kind of guiding them in how to move forward. So yeah, it's basically us standing back and being supportive. So those kind of things I've been doing. It's almost certainly not enough, but you know.
More questions?
I don't know if you would be able to answer this question, but you said in your talk at one point you would have put the cap on protein preservation at 1 million years.
Yeah, exactly.
And you talk about dinosaurs.
Yes.
If someone were to come to you now and say, where is the upper limit? Can we even assess where the upper limit is? Or would you...
Well, the thing, of course, you have to think about that in terms of where in the world you are. But I would have thought if you're in the high Arctic, 30 million. I think if you're in Antarctica, because it hasn't moved as much, you may be able to go back to about 40. But that is in tooth enamel. And what's really interesting about tooth enamel is different species decay at different rates. And so if you've got something which is evolving and decaying more slowly, and we don't know why, then that could go back further. And I would not be at all surprised. But it would have to be somewhere kind of special. The bottom of the sea would be a good place to go back beyond the last extinction event, the big, the KT boundary. But I think that would be, you're talking about, the obvious one I've been talking to lots of my colleagues about, we're getting very excited about is shark teeth because they shed them so much and they're in marine sediments and they're cold. So that's a place that I know, I'm chatting with a lot of young scholars and they're really interested in that. So my mind would be, if we're going to beat that limit, it's going to be in shark teeth. But I don't know is the answer, but I wouldn't, if I heard there was a great sequence came out of shark teeth from the Cretaceous, I wouldn't lose sleep over it anymore. I would have just dismissed it 5 years ago. Yeah.
So, you were talking about DNA, and now we talk about proteins, and my question is, where do you think, or you would like the proteomics research go in the future? So we were really focusing with DNA in our field, in paleontology, in the taxonomic utility. does this have to be the same for proteomics? We should expect that we should use other different type of applications. What do you think should be the future of this field, or what you would like it to be?
So there's a whole series of things that I would like to look at. And one is the unification with stable isotope research. Because in theory, we're getting close to a point in which we can sequence a peptide mass spectrometrically, then analyze the individual isotopic composition of the amino acids in that peptide. And that will be mind-blowing, because then we'd actually get a metabolic story as well. So that's one thing I think will be really, really interesting. The other one that, of course, I would say is diagenesis, because I think that gives you a whole window into geochronology as well. So that, I think, is extremely important. And then what I will be saying is that it's a blunt tool. It's a blunt tool, and unless you get a really rich proteome. But what's extraordinary about the Harbin proteome is it's really, really rich. And so, and yet it seems that it's not giving us as much information as I would have hoped it would have given us. And that's not going to be good news for other samples. Harbin should have DNA in it. I mean, I think more Harbin samples are going to have DNA in them, and then the DNA will tell the story, not the proteins. So I think that with the proteins, it's, if you really want to look at, let's say, biological sex, then it could be potentially great for that, but you have to make sure it's working in your system. Chronology is great. And then there's the other kind of boring thing that you guys are never going to do, and we've done a lot of these already, though. It's where you have that, where does this taxonomy fit? I don't know where this thing fits. And there you certainly can if the split is deep enough. But I've been sitting on a very, very rich data set since 2017, 2018, on a 3 million-year-old bear sample, so a very rich proteome. It's not published because I don't think we have the software to analyze it correctly, and if we use it at the moment, we cannot tell if it's the base of all bears or a side branch, and yet we have got a massive amount of proteomics data. It's just, it was too shallow. I mean, the carnivores have not evolved fast enough, and it wasn't a question that could be resolved, even with the wonderfully rich proteome we've got. So there's no real taxonomic story, which was the hope. It's going to be a story about how you tune software to get the most out of the data. And I think that's been the problem, is kind of this naive assumption that we could get a lot of phylogenetic information out. And the realization it's going to be much better for a whole load of other things. And I think that if you want to do Denisovans, I would use zooms. I wouldn't use deep sequencing, because if you've got a mass shift and it's real, you'll see it.
Thank you.
Thank you. I was wondering if we could elaborate. There is a topic that you mentioned in the last slide, which is proteomics or proteins on stone tools.
Yes.
Elaborate a little bit on this.
Absolutely. So this is something which I think definitely won't work. But as you see, I'm not very good at predicting. And so if you're working, so for my mind, the protein would need to be protected from exoenzymes produced particularly by bacteria, because bacteria know that proteins are sticky on surfaces, they have surfactants, they have enzymes that work by being pushed out into the environment and degrading things. So you'd have to protect against that. But those of you that work on lithics, and I don't work on lithics, is if you're developing a polish on the lithic, then you've actually got a mixture of fine-grained minerals and potentially some proteins. And if that relithifies, recrystallizes, maybe. And so what I don't like is that people have been saying that you can take a lithic artifact, you can put a drop of water on it and extract proteins. Really? It's been in the ground for how long? With water flowing through it, and yet you could just put some water on it. But and this was interesting about this new technology, is it's actually a more aggressive and thoughtful way to recover proteins. And what's also interesting, in the old days they were using antibodies, and they were guessing at things like immunoglobulins and serum albumins. these proteins they're getting off these lithics, and it's unpublished data, and I've not reviewed it, I just helped my postdoc to write the review. They make sense, and it's like, that might be quite interesting. And interesting that the point was, the team that published it will not release the raw data. And I was saying, like, no one will ever take any interest in your work unless you release the raw data and allow other people to analyze it. But if they do, if they will or not, I don't know, and we can all kind of clamber over it. Like the dinosaur stuff, I mean, I'm not convinced by the recent dinosaur paper, because when I've looked through the raw data, something doesn't seem right to me, but I need to mine it more deeply. But again, I think if that was the case, that would be amazing, right? And then we could start looking at all this artifact use. So that's what I would say. I would not even have mentioned it 2 months ago. But now I think we have to think about how that might be possible. What circumstances might you get proteins which are very exposed and trapped on a lithic surface? And that kind of rock flour that gets generated by use, maybe. Working a bone, I mean, you've got some calcium phosphate coming off. So yeah, I don't know is the answer. But I think it's something to keep one's eyes open about. Because definitely we get proteins in mortars, we get proteins on lots of different surfaces. Proteins are really sticky. The problem is then bacteria know that, and they're very good at eating the proteins which are stuck on the surfaces. So you'd have to think of what would the mechanism be to inhibit the microbes. But, I mean, we've been looking at proteins on pottery, and honestly, those proteins are impossible to get off. In the end, we've been hydrolyzing the pots with strong acid just to measure that, yes, there really were amino acids on that pot, we just can't extract them. So they're sticking really tightly. So maybe.
So thank you very, very much. I'm sure that there are a lot of questions and people are really looking forward to keep talking to you, because we're going to have now a cocktail or something to have the chance to enjoy and celebrate. And also before you leave, we just want to give you a little gift for you to remember your visit here. For us, it's really a very special occasion, but it's light, okay? So thank you very much, Professor Kohn.
I don't know.